Myself, Divya Shanmugarajan, on behalf of Altum Technologies, I welcome you all for joining us today in this last but not least sessions of Women's Day special webinar series. We are excited to have you all here. We all know that today is the International Women's Day, and so I would like to welcome all the women out there. And on behalf of Altum Technologies, I wish you all a very happy and International Women's Day. It is a day to express our gratitude to the women in one life, both personally and professionally. And Alton Technologies salutes all the women out there who are not we are just doing wonders around the world. It won't complete without expressing my gratitude to each of you who have contributed this Women's Day special series webinars organized by Alton Technologies. And let's start today's sessions. I told it's the last session, but certainly we are coming out with the new sessions in near future. Today's session is going to be very interesting. It is unlocking a drug discovery. What is unlocking drug discovery? You might have been more anticipated to hear from a speaker, certainly. So the today's topic is going to be on unlocking your drug discovery, navigating through homology modeling and pharmacophore modeling for targeted therapeutics. We all know that we are living in the AI century where the computational tools play an essential role in drug discovery and development. Without introducing your computational techniques, it's very well known that the drug discovery certainly will take more than 16 to 20 years for any one drug come to the market. When we try to introduce these kinds of technology, it not only booms your research and also saves your time and more cost-effective manner. So I think you all feel more excited to hear from this. Come, it's time to introduce our speaker of the day, Dr. Prabitha P., who is currently working as a lecturer in Pharmaceutical Chemistry Department of JSS College of Pharmacy, Mysuru. She obtained a PhD degree, MSc degree, and postgraduate diploma in computer-aided drug discovery from an JSS College of Pharmacy, Mysuru. Her primary research is key areas are synthesis of small molecules for neurodegenerative diseases, and she is skilled with the various techniques like analytical techniques, cell culturing, and in vivo studies, computer-aided drug designing. To add her accolades, she has got a lots of publications in a both national as well as in an international publication. So I think you will be so excited to hear from her. Without any further delay, let's start the session and hand over the session to Dr. Prabitha ma'am. Please take the session. Hand over this platform to you.
Yeah, thank you for the warm introduction as well. So greetings to all. So first, before I start the session, at the outset, I would like to thank the organizers, Altum Technology, for granting me a chance to speak in this special Women's Day webinar series. So thank you, Divya ma'am, for giving me an opportunity. So it's actually a very great feeling to speak on such a big forum like this. So before I get into the presentations, I would take this opportunity to wish all a happy Women's Day to all the beautiful ladies out there. So all our smart ladies. So today's era, we are the rulers actually. So it is great to be part of this webinar series. So looking forward for more such events to come. Thank you, ma'am. So
yes, certainly.
Shall we start the presentation?
Yes, certainly. Yeah. Please move on. Thank you.
So if we look into the evolution landscape of drug discovery, the integration advanced computational techniques plays a pivotal role in accelerating the identification and the development of novel therapeutics. So I'm here to discuss the synergistic power of two computational approaches, homology modeling and pharmacophore modeling, that has become indispensable tools in the drug discovery toolbox. So as we all know that homology modeling is considered to be a most accurate computational method for structural prediction. So it is one of the computational structure predictional method that is used to determine protein 3D structure from its amino acid sequence. That means it predicts the protein structure based on the sequence homology. So here it is also known as comparative modeling because it uses a key template related 3D protein structure to generate the model for the target protein. So this technique has proven valuable in molecular biology, discovery, and research. So prediction of the three-dimensional structure of a given protein sequence is nothing but it takes the amino acid sequence from a homologous protein or template protein for which an X-ray and an NMR structure is available, and based on the alignment of one or more structural proteins. So if the similarity of the target sequence and the template sequence is detected, it is assumed that the structural similarity can also be obtained. So in general, for a model to generate, there should be at least a 30% sequence identity. But it is said that if higher sequence identity is there, then the model accuracy is obtained. So when you see, homology modeling basically depends upon two principles. One is the primary sequence of amino acids that determines the protein 3D structure, and the other one is the protein 3D structure is somehow conserved with regards to the primary sequence. So homology modeling is considered as a powerful approach to generate models. So, why homology modeling is? It is useful in all situations where the structure of the protein is required. Example, if a structure-based drug design, if you take a structure-based drug design, a protein structure is used as a basis to identify or design a new chemical compound that could bind to the target, resulting in inhibition of the target protein. So in such cases where the protein structure is actually necessary to identify new chemical entity, this homology modeling is very helpful. And also it can be done only there is a requirement when experimental structures of one or more homologous proteins exist. That is, when you have an NMR or a crystal structure, X-ray crystal structure, only from that sequence, taking it as a template or reference, this homology model can be built. And also, one great advantage is useful when these experimental methods like X-ray crystallography or NMR techniques are difficult and time consuming, and also it is expensive to use. In such cases, homology modeling is one of the computational approach tool where it reduces the cost, so low cost and less time consuming. And that is the reason why people tend to have these in silico tools as a powerful approach for drug discovery process. So when you see it to the homologous proteins, so the sequence homology modeling basically relies on proteins with sequence similarity, and alignment of the target and the template sequence is the one of the important and the critical step in this homology modeling, because I told you that sequence identity is good, then only the model accuracy will be generated. So that is one of the major impact of this homology modeling. So alignment, initial alignment, alignment correction, these are the things which has to be taken into consideration when you generate a good model. And the other part is prediction of this confirmation prediction. That is, from this homologous proteins we can predict the 3D tertiary structure, or nothing but the 3D structure of the protein. So these are the important things we have to keep in mind when you start to model a protein. So, sequence similarity and structural similarity is a very important aspect in homology modeling. As long as the length of the two sequence and the percentage of identical residues fall in the region of safe zone, the sequence are practically guaranteed to adopt a similar structure. That is, what is meant is, if there is 90% sequence similarity, then between the template sequence and the unknown sequence, then it is understood that the model predicted will also be as similar as the template sequence. So the model accuracy basically depends upon the identical sequence. So higher the identity sequence, greater the efficacy or the or the model generation will be powerful or accurate. So basically, the concept of homology modeling is, if there is an unknown structure, 3D structure, we have a template, then through sequence alignment, we generate a 3D structural model. That is the basic concept of homology modeling. So how there is a steps involved in this homology modeling. So it goes with the flow. So you have to initially take the sequence of the target, then select that template. So the best template is selected first, then alignment of the target sequence and the selected template sequence. And then homology model is generated using different tools. One such tool used in Discovery Studio's Modeler. And later, after selection, assessment and refinement of the best homology model is being done, carried out. So this is how the workflow of the homology modeling takes place. So I've already told template recognition and initial. So initially, the sequence will be initially a blast will be performed. That is, alignment of protein sequence will be done initially, and the initial alignment and template recognition is the first step in homology modeling. Later, whatever aligned sequence, if there is any gaps or any irregularities, that will be corrected in the alignment correction stage. Later, the backbone generation, that is, the coordinates of the protein sequence, is being done, followed by loop modeling and sidechain modeling, and then model optimization. So, if the model is not having a good or low percentage accuracy, then this process is repeated until the best model is generated. So, validation of the model is basically done in homology modeling through Ramachandran plot. So, when you see this, the best homology model, you can see, we can visually see that Ramachandran plot before refinement, how it was. That is, certain non-glyc, the 95% of the non-glycous glyc residues should be in the favorable regions. So that is why we tell that Raman model validation is basically done by Ramachandran plot. After refinement, we can visually see that some of the amino acids which was in the unfavorable region has pulled over to the favorable regions, like iteration processes. So the template selection has certain functionalities. That is, one is some sequence identity. I told you higher sequence identity will improve the of the template, will improve the model accuracy. So that is the main principle behind this homology modeling. So sequence identity with the template should be there, only then the model accuracy will be obtained. Next is a well resolved and high quality structures will make a better template. And functional similarity, that is, the template should have similar functions to the target protein. That is how the sequence similarity will be obtained. So when you see, I told you homology modeling basic step is template recognition. So how to get this template recognition is, the query sequence is obtained, will be put in the blast server of the web, and that will search a list of hits, the modeling templates and corresponding alignments from the protein data bank. So if you see, blast is nothing but it is the most widely used bioinformatics programs. It is called as basic local alignment search tool. It emphasizes speed over sensitivity. So from the huge genome database, the algorithm is taken up and then it is put into the blast server. So this is the work window of the blast server. So here we have different blast databases, nucleotide blast, protein blast, any type of blast can be done. So the sequence is put into this and the blast run is done. So first, a blast has been so blast is nothing but a pairwise alignment of protein sequence with all other sequences present in different database, like the database present in Swissprot, Uniprot. So highly identical. So after the blast is performed, the highly identical sequence is obtained. The identical sequence should be very similar to the unknown sequence. Then the 3D structure of the protein is predicted based on the structure of highly identical sequence. So basically, homologic modeling is based on the knowledge-based method. It is called as generally a knowledge-based method because it is based on the existing protein structure information. There is one more, apart from homology modeling, there is one more knowledge-based method which is called as threading. Threading is also similar to homology modeling, but in homology modeling determines structure model based on the experimental structures determined closely related to the sequence. So based on that only homology modeling is generally generated. But threading is also similar to structural similarity, but here we can use a detectable sequence similarity can be used or can be detected or need not be detected. That is, with or without detectable sequence similarity, threading can be performed. So that is the difference between homology modeling and threading. So in homology modeling, identical sequence is very important, but in threading, structural similarity can be predicted, but there is no need for a sequence similarity. So the alignment, the initial alignment, the residue exchange matrix and all is taken into steps. So these are the steps which is initially important while doing a homology modeling. So the best thing is the UniProt or the SwissProt. These databases will give the sequence, protein sequence for the target protein. So you have to put the query into the blast, get a high identical sequence, and then predict the structure based on the high structure of the highly identical sequence. So these are the initial steps which is involved. And then alignment matrix. So by comparing thousands of sequence and sequence families, it has become clear that the opening of gaps is about as unlikely as at least a couple of non-identical residues in a row. So for every gap, if you see the next slide, uh, alignment matrix I've shown before, that alignment matrix plays a very important role. That is, the initial template recognition and alignment matrix happens in the homology modeling step. So for every the residue, the gap has been aligned, and then alignment correction has been done. Sometimes it is very difficult to align two sequence in a region. So when in that cases, we can use another sequence from the homologic protein to find a solution for this alignment sequence. So if you see this, so a model with a larger gap will not be taken into consideration. A model with a small gap or aligned, that is, as much as possible the sequence alignment is necessary for the model to be considered. So after that, the template and the sequence, target sequence and the template sequence is checked for the alignment corrections. Later, once the alignment is corrected, the backbone generation has taken place. So one can simply copy the coordinates of the template residues that show up in the alignment with the model sequence. So basically what happens, if the two aligned residues differ, only the backbone coordinates, that is C and C alpha n, can be copied. If they are same, we can also include the side chain and functional group. The side chain and functional group is the add-on. So if you see peptide chain and all, they have a specific sequence. So backbone model building is very important step in homology modeling, because the template residues can be aligned with this model sequence. Next is loop modeling, or we always tell as sidechain refinement. This is also based on two approaches. One is on the basis of the knowledge of the PDBs, which has so for known loops with endpoints, we will match the residues with this loop, and they're simply the loops will be copied from the template to the new sequence. And based on the energy function also, the loop is being judged, the quality of the loop is being judged. So basically the energy function used here for model optimization is ab initio fold prediction. Then sidechain modeling, I told you, sidechain modeling is sidechain refinement, we usually tell. So it is therefore possible to simply compare. So I told you the principle behind homology modeling is the protein 3D structure is somehow conserved with regards to the primary sequence. So it is therefore possible to simply copy these conserved residues entirely from the template to the model. So model optimization, that is the energy is been taken into consideration. So it is model refinement is usually done by energy functions. If suppose that a model is validated that there is no irregularities or there is no steric hindrance between the amino acid residues, those has to be taken up into consideration before the model has been refined, and then model has been evaluated. So correctness of the overall fold structure and the stereochemical parameters like the bond length, angles, etc., also has to be taken into consideration for the model validation. So if you see, model validation usually is done by the Ramachandran plot here, where at least 95% of the non-glide residues should be within the allowed regions of the plot. So residues in the disallowed regions we will should be checked, and the reason for that also should be explained. So why that certain amino acids like histidine or valine is out in the unfavorable or disallowed region, that also has to be taken into consideration. And in some cases it may be necessarily to explain why that disallowed regions is being occurred. So these are the basic workflow. So starting from the template selection, sequence alignment, backbone model building, loop modeling or sidechain refinement, then model refinement using energy function, and model validation. So this, there are 7 steps of involved in this homology modeling. So to build a good model, the that's the reason why homology modeling is considered as a powerful approach to generate model, and it is also been considered as an accurate method for structure prediction tools. So when you see the application in drug discovery, there is various applications. One is target-based drug design. The other one is protein ligand interactions. So homology model basically is nowadays it forms the cornerstone of rational drug design therapy, and it's also modeling ligand binding assist in studying the protein drug interactions. So protein ligand interactions can be studied, and also it can be used to understand design mutations, identify the clefts and cavities that may function as active or binding sites, and understand their functions. And also it is possible to identify the conformational epitopes, that is nothing but the neighboring amino acid residues located on the antigenic protein surface structure can also be predicted or identified using this homology modeling technique. So when you see the future developments in this technique, so as ma'am told in the initial that computational tool has been very well correlated with the machine learning or artificial intelligence. So enhancement in this modeling softwares will also be used for more accurate predictions, and also advanced 3D imaging techniques can also be used. Advanced imaging techniques to elucidate the 3D structure of biological molecules, which is otherwise called as cryo-EM, can also be applied in homology modeling, and also this model can be used for targeted drug development. So in recent advancement, targeted homology model is being aligned with the artificial intelligence or machine learning concept, where there are some algorithms used to enhance the model software for accurate predictions. So this is all about homology modeling. So after COVID, there are if you see in the recent after COVID, there are several articles which have come out related to homology modeling. Um, so, homology modeling has served as a very important tool in drug discovery in the recent times due to its more accuracy predictions and the utilization of the targeted drug development. So, that way, homology is modeling is one of the important and indispensable tool in the drug discovery process. So this is all about homology modeling because it is in recent times as considered to be one of the most accurate computer aided prediction tool. So next I would like to talk about pharmacophore mapping on pharmacophore modeling. It is also one of the computational technique used in drug discovery. It has wide range of applications. The When you take pharmacophore modeling, as per Ehrlich, pharmacophore modeling is he was a scientist who coined the term pharmacophore. It is nothing but the molecular framework that carries the essential features responsible for a drug's biological activity. So, in short, in the recent times, pharmacophore modeling is defined as an ensemble of steric and electronic features required to ensure optimal supramolecular interactions with the specific biological target to trigger or block the biological response. So In order to identify and optimize molecule that bind to a specific target, we must understand the essential molecular features of pharmacophore that is necessary for the molecule to interact with the targets. So usually the molecular interactions between the ligand and receptor are represented by a set of pharmacophoric features such as hydrogen bond donors, acceptors, aromatic rings, and hydrophobic regions. Uh, these pharmacophoric features are used to create a three-dimensional model of the binding site, which can be used to identify the molecule that are likely to bind to the target. So, this is one of the important tool for understanding the protein ligand interactions. So, there are different types of pharmacophore mapping. One is ligand-based pharmacophore mapping. So, this approach uses the structural and physiochemical properties of a set of known ligands that bind to a target to identify the common feature, which common pharmacophoric feature. So, whatever model it is generated, that model can be used to screen the library of compounds for molecules with similar properties and potentially identify the high binding affinity molecule. So, this is one such approach where the ligand's binding property is being taken into consideration. The next approach is receptor based pharmacophoric mapping. This approach involves generation of a pharmacophore model based on the 3D dimensional structure. So here the protein structure, the active site amino acid residues are taken into consideration. Uh, for generating the pharmacophore model. So if you see this approach, it is basically that is the three-dimensional structure of the target receptor which can be obtained by X-ray crystallography, NMR, or homology model. The model can be used to design or screen the compounds that interact with the receptors. So, if you see here also the active site amino acid residues pharmacophore features is being taken into consideration. Based on that, the ligands are designed so that there is a better binding affinity between the ligand and the receptor. The next is hybrid pharmacophore mapping. It is, as the name suggests, it is the combination of ligand and receptor based pharmacophore model. It takes advantages of both the types of informations. It is basically used when the three-dimensional structure of the target is not available, or when the ligand binding is poorly defined. Ligand binding site is poorly defined, which we tell as active site. When it is poorly defined, we can use this method to find out the pharmacophoric features which is essential, or a model can be generated between the target protein and the ligand. So this is a hybrid pharmacophore mapping technique which can be used in virtually screening the data sets of ligands. The next approach, which is a well-known approach, is 3D QSAR pharmacophore mapping. This approach involves the use of quantum mechanism and dynamic simulation to generate a dynamic time-dependent pharmacophore model. Uh, Basically, it is accounts for the flexibility and motion of both the ligand and the receptor. So it basically gives an or provides an insight about the molecular mechanism of the ligand receptor interaction, and it is used to design compounds with specific properties or mode of action. So this is a well-known method, and I think the generated model has more accuracy when compared to other methods. 3D QSAR pharmacophore mapping method gives a well-defined pharmacophore model. The other type of pharmacophore mapping is fragment-based pharmacophore mapping. So not only ligand-based, when to screen a large library of compounds, this method is also used. This approach involves the identification of the key functional groups of the fragments that are important for the ligand binding and optimizations. So the fragments you can assemble into a larger compounds with the desired pharmacophoric features and properties, and this is basically useful for designing drug targets with improved properties, potencies, and selectivity. So pharmacokinetic properties can be included when we use this type of approach. When you know that fragment based drug design is one of the important aspect of drug discovery, that in order to design a candidate molecule with improved potency, this approach can be used. So the process of deriving this pharmacophore is called as pharmacophore mapping. So first, in the pharmacophore mapping, they will identify the common binding element that is responsible for biological activity, then generate a potential conformation that active compound may adopt, and determine the 3D relationship between the pharmacophore element in each conformation generation. So the predictability of the result, in vitro result, will be very good when we design such type of molecules using this pharmacophore model approach. So basically, this pharmacophore model uses conformational approach. So conformation generally means structural arrangement. So the torsion angles, the bond angles, and the different ring taken into consideration, and the biological activity of this molecule will be strongly dependent on the conformation. So it is basically done by exploring the energy surface of the molecule and determining the conformation with minimum energy. So conformational analysis is needed to identify the ideal conformation of a molecule. So this conformational search is a very important tool in pharmacophore model generation, because it is needed to identify the ideal conformation of a molecule. So when you see the applications of pharmacophore modeling, it is used in various stages of drug discovery, starting from lead identification to optimization to drug design, virtual screening, scaffold hopping. So, in lead identification, it can be used to identify the lead compounds that possess the desired pharmacological activities. So, you generate a model. That model can be used to virtually screen the database, or de novo drug design can also be used. Then multi-target drug design, activity profiling, target identification can also be performed from the generated model. So lead optimization is also be, that is, by identifying the key structural features responsible for the desired activity, we can optimize the lead compound. So from the database we get a lead, then we uh, take it for the in vitro study, understand the potency of the drug, then take up again get in the lead compounds, optimize the lead compounds to identify the key structural features responsible for the biological activity. In the drug design, so if you want to design a new drug by identifying the key structural features, this method can be adopted. Even when you want to screen a large database of compounds to identify the potential hits and the desired pharmacophore, this method can be adopted. Scaffold hopping also, this method can be adopted because pharmacophore mapping can be used to identify new scaffolds that can be used to develop compounds with desired activity. So there is a vast application in the field of drug discovery, starting from hits to lead optimization, virtual screening, scaffold hopping. Every part of the drug discovery, this method is being adopted. So when you see the identification of application of this drug discovery, binding site identification, that is, pharmacophore mapping helps in the identification of the binding site of a protein or receptor. So the generator pharmacophore modeling, using that we can uh, design the ligands with desired activity based on the binding site pharmacophore features. That is, structural activity relationship can also be analyzed using this pharmacophore data. So pharmacophore mapping can be used to analyze SAR data to identify. So basically, the electronic and the steric features which ensure the optimal supramolecular interactions with the specific biological target can be analyzed, so that it can be used to trigger or block the biological response. So the essential features for biological activity can be determined by pharmacophore model. So using different techniques, like starting from ligand based, receptor based, fragment based, 3D pharmacophore, QSAR based, pharmacophore mapping can also be used. Fragment based drug design, I told you initially that it identify fragments that can be used to build larger molecules. Uh, so basically these computational tools have been used in such a way that the efficacy and the potency of the desired candidate molecule is improved. Then toxicity prediction can also be used to, so that we can understand which are the key functional groups which is responsible for generating the toxicity in the wet lab. So the admit toxicity profile can also be taken into consideration, can be predicted by this pharmacophore mapping features. So it has some limitations. The accuracy of pharmacophore mapping is limited by the quality and quantity of available experimental data. So sometimes, due to the incomplete data, it can lead to generation of inaccurate pharmacophore models. Basically, the pharmacophore model is based on the fit value. So better the fit value, greater the model generator. And overfitting can also be happening, so resulting in poor performance when applied to a new data. So that's the reason why I told you 3D QSAR pharmacophore mapping is one of the prominent and promising tool to be explored when designing, or when you want to identify and optimize the new leads. So that tool can be used as one of the essential parameter in screening the database of compounds. Complexity of targets, so some targets such as protein protein interactions or membrane proteins, it is very difficult to model the pharmacophore using pharmacophore mapping due to the complex structures and dynamic nature. So these are some of the limitations, but when it comes to small molecule generation of leads and lead optimization, pharmacophore mapping is one of a powerful approach or proven approach to generate a model, and can be used in the drug discovery as an indispensable tool. So these are some of the parameters which I wanted to discuss. So if any questions related to this, I'm free to answer. So when you see into the drug discovery process, homology modeling and pharmacophore modeling plays a very important role. So these tools can be used. Apart from that, molecular docking is one of the other tool which is generally used for drug discovery approach. So screening a large database, I would suggest that you can go through these tools where you can pharmacophore model prediction and then get into molecular docking. The accuracy of the results will be very good.
Yeah. Yeah, thank you so much, Dr. Prabitha, for giving a more insights on various approaches towards the homology modeling and as well as the pharmacophore modeling. So to add to that, we have questions are there in the platform. So we will you can see the questions, right? Can you see the
It's in the
questions?
chat box, ma'am.
No, ma'am. It is just displaying in a screen. Can you see that?
Okay, how homology modeling accelerate drug discovery? So, I told you homology modeling is basically if there is no structure available, 3D structure available for the protein, uh, we use this homology modeling technique to actually get the 3D protein structure using a template sequence. That is, a sequence homology is required for generating a protein structure. So accelerate drug discoveries in the recent times when you take into consideration in the COVID. So SARS-CoV-2 doesn't had a 3D protein structure. So finding the 3D protein structure through experimental methods will be more expensive and time consuming. In that case, we can use these tools to predict the 3D structure, tertiary structure of the protein, and then design the ligands and screen those ligands into the target protein and understand the binding interaction between the target protein sequence and ligands. So that's how, at low cost, and if I say in one word, it will accelerate the drug discovery process because it is a time consuming process and a very reliable and accurate computational approach.
Yeah, I would like to add towards that, and we have in a Discovery Studio the modular algorithm,
Yes,
which is
modular
known
algorithm.
for more than 3 decades. which is it's a part of Discovery Studio, where you can model the structures more accurately. I believe that during the COVID times, many people
Yeah,
utilize this
yes,
kind
yes.
of technology and did a modeling and come out with a good number of leads to that. Yeah, thank you, ma'am.
Yeah, yeah.
And
So lead optimization was done using homology modeling. So that was a great achievement in the field of.
yeah, I think you have one more
So
question.
how
So
structure-based pharmacophore is important then? See, in ligand-based pharmacophore, we need not know the pharmacophore features of the protein structure. So basically, in ligand-based pharmacophore mapping, what is done is both when you take in one or the other, it is very important. So in structure-based pharmacophore, the structure of the receptor, the pharmacophoric features of the receptor is taken into consideration. In ligand based, the ligands, not just one or two ligands, when there is a large database of ligands, to understand the common feature pharmacophore essential for binding of the ligands to the target receptor is taken into consideration. So each is a different type of module, but both understands and explains the protein ligand interactions, so that it gives an high affinity in a type of interaction between the protein and the ligand. So which are the pharmacophore features essentials for the ligand to bind to the protein, and in case of structure based pharmacophore, the structural features of the active site amino acid residue is taken into consideration. So if you see, structure-based pharmacophore more is more important than ligand-based. I feel both are very important. They
that's
do
true,
carry their
actually.
own weightage.
Yeah, so
So
both
it is
should
not
work
one
together
better
to
than the
get
other.
an best results, I believe. So yeah,
Yes,
thank you.
yes. Both
Thank
have
you.
their own results.
And okay, can all types of proteins can be modeled? I think it will be very challenging for you.
Yeah, yeah. Uh, see, a human genome database, all the information is obtained from these databases. So I think when we see, generally, I told you there should be an homologous proteins existing when you are doing homology modeling. That is, there should be a reference sequence. Only based on that we can create an. That is, there should be an template so that we can form a base. Without no base, then we cannot model any protein. So that is what I feel. So if there is an NMR or an X-ray crystal available, which has an experimental method, through experimental method those sequence are being obtained, then that can be used. So for homology modeling, the basic is it should have a sequence homology. So based on the sequence homology only, the protein or the tertiary structure is predicted. So there
Yeah,
should be an sequence which is, so all
yeah.
type of proteins cannot be done. But yes, I think ma'am, you can correct me if I'm wrong,
Yeah,
nucleic
certainly. Yeah, any types of proteins, as Dr. Prabitha was mentioning, you should have some kind of template identification should be there, and homology. That's why we call the homology modeling. And if
yes,
you have any types of proteins, so as a question is asked, any types of protein can be modeled when we know the templates.
templates.
So when you know the template, certainly your results of a drug discovery also will be accelerated. Yeah, thank you, Prabitha. And
Thank you, ma'am.
again,
Which
the
type
question
virtual screen you prefer for better screening of
compounds.
which type virtual screen you prefer for better screening
I
of
believe
compounds?
that if I rephrase this, which type of ritual screening method is better for screening the compounds?
So I meant to tell homology modeling or pharmacophore modeling, right? So
Which type of ritual screening they
if
are
you
mentioning?
see, initially when you're starting, I feel that pharmacophore modeling is a better tool for designing a new ligand, because you have an idea about the protein structure, the target protein, which has a 3D structure. I'm talking about the co-crystallized ligand or an protein which has a tertiary structure that can be acting as a receptor, and the ligands can be used. So initially when you're starting a drug discovery process, you can try with pharmacophore modeling, because the you can understand what are the features which is essential for getting into the protein ligand interactions. So mm,
Yeah, I
for
think
better screening of compounds, a large database of compounds, pharmacophore model method is a very reliable method of.
yeah, yeah. Thank you so much, Prabitha. I think there is
Any
one more question.
conditions to collect compound for 3D QS? Yeah, pharmacophore. basically it should have an experimental value when you're doing 3D QSAR pharmacophore. Uh, if more the number of compounds, better the activity model generated, the prediction of the model will be better. And it is very challenging because if you take experimental method with an IC50 value, you should ensure that the same assay method is being utilized in all the compounds you have collected, so that the predictability of the model will be accurate. So that is little challenging when you take to 3D QSAR. But 3D QSAR, since we take a large database of compounds and then screen that with an experimental result, so the accuracy of the model generated will be more. Uh, that is what I felt in 3D QSAR. So any condition is, you have if you have a good number of compounds with an experimental result, IC50 value, with preferably same assay method, so that
Yes,
uh, the predictability of the model will be good. So
that's
that
true.
is
That's
one
true. Because,
condition to collect
of
compounds.
course, because that is a very big challenging in the
Yeah, big
QSAR,
challenge.
QSAR pharmacophore.
Case of 3D QSAR, that is a very big challenge actually.
Data mining is a toughest process, but the remaining process, I believe it will be good. And then I think we have come to an final session. I think it's very informative, and you can see over the commands has been given by
Yeah,
the
thank you, ma'am. Hope to.
and we will never leave you by this. And it
No,
is a
sure,
time to
sure. I will be in
felicitate
touch with
our
you.
speaker and. So thank you so much, Prabitha, for joining
Thank you,
with us
ma'am.
in
Thank you.
on a wonderful day of International Women's Day celebrations.
Yeah, it was
And
a memorable day for me too. So
yeah, it's
thank
our
you for that.
pleasure too. Yeah, it's our pleasure too. And to put together, I think we have invited 8 different women of different backgrounds to present it. And I take this opportunity. I extend my heartful gratitude to all the women scientists from academia who presented their invaluable sites on computational approaches in next generation therapeutics, agriculture informatics, network pharmacology, and today's speaker was so strong in the pharmacological and pharmaceutical chemistry on the Altum platform. I believe that your dedication, expertise, and innovation contributions are the driving achievements for the young women's. And your achievements towards your crucial fields is shaping the future of science and benefiting the whole society. And it is not but not least, I'll tell you that your passion towards a research commitment to excellence, you are aspiring, you are, and you people are trying to be epitome for your various streams by sharing your knowledge and expertise, and also you are fostering your collaborations and exchanging your knowledge of the scientific community, all places significant contributions. And thank you for your perseverance and leaderships, and your works are instrumental in addressing a more precising challenges in your various fields of drug discovery and development of computational biology and chemistry. And I wish you all your true effects is really appreciated. And finally, I would like to thank all the women scientists who present in the Altum platform as an empowering, and also I will tell that you all are great people to given shaping the future scientist actually. With this, I thank you all once again, and thank you the today's speaker and then previous
Thank you.
speakers, and I wish you all success from the Altum Sen. Thank you all.
Thank you, ma'am, and wish you a happy
And
Women's
happy
Day again.
International Women's Day once again to everyone, and thank you all.
Thank you. Thank you.